covers chapters 7, 8, 9, and 10. Chapter 7 was how cells harvest energy from food. Chapter 8 went over mitosis. Chapter 9 went over meiosis. And in chapter 10, we discussed the foundation of genetics. So, in cellular respiration, which we talked about a lot in chapter 7, we discussed how eukaryotes produce most of their ATP by harvesting electrons from the food molecule glucose. Oxygen is used to transform glucose into usable energy, and the energy invested in building the organic molecules is retrieved by stripping away electrons and using them to make ATP. So here you can see the formula that is the basic breakdown of the formula that is happening during cellular respiration. We have our glucose molecules and our oxygen as reactants. We are producing CO2, water, and energy in the form of ATP. The reactants are glucose and oxygen. The products are carbon dioxide, water, and energy. Glucose is transformed into energy during cellular respiration through 3 different processes. We talk about them as if they're happening in chronological order, but really they're just a continuous cycle that are constantly feeding into each other. But for the sake of understanding, our first part of our cellular respiration process is glycolysis. This is where we are breaking down our glucose molecule into pyruvate in the cytoplasm of the cell. So glycolysis is happening here. We are within the cell in the cytoplasm. In the Krebs cycle, the second part of this process, we are harvesting energy from pyruvate that was transformed into acetyl-CoA in the mitochondrial matrix. So the Krebs cycle is happening in the mitochondrial matrix. You can see here. But before that pyruvate is usable, it needs to become acetyl-CoA, and that happens in a transition step between glycolysis and the Krebs cycle. So once pyruvate has been transformed into acetyl-CoA, it is usable for the Krebs cycle. Then we can go on to the electron transport chain, and this is where the majority of ATP is made, and it is also happening in the inner mitochondrial membrane, which is within the mitochondrial matrix, or so also surrounding the mitochondrial matrix. In cellular respiration, you can see we are making ATP throughout each part of the process, but the majority of the ATP that we are producing is coming from this last step, the electron transport chain. We have 2 ATP that are produced in glycolysis per 1 molecule of glucose, 2 ATP produced in the Krebs cycle per 1 molecule of glucose as well, and that same molecule of glucose, once its components have made it to the electron transport chain, will produce a whopping 34 molecules of ATP. So you can see here is that transition step where pyruvate is being transformed into acetyl-CoA so it can be used in the Krebs cycle. So electrons pass from atoms or molecules in many chemical reactions, and those include the ones in cellular respiration. So these reactions are called oxidation reactions when an atom or molecule is losing an electron. And the reactions are called reduction reactions when an atom or molecule is gaining an electron. Excuse that typo, but this should also say electron. These reactions always occur together. Here we have an example. You can see in our oxidation, we are losing an electron for a product that has a slightly more positive charge because we lost a negative charge electron. In our reduction, we are gaining an electron, so our product is going to have a slightly more negative charge after this reaction. In many redox reactions, oxygen is a common electron acceptor, and in cellular respiration, it is the final electron acceptor in the process. Energy follows the electrons such that the reduced form of an organic molecule has a higher level of energy than the oxidized form. So our reduced form will have a higher level of energy than the oxidized form. When we're talking about glycolysis, this is the breakdown of glucose. It is taking place in the cytoplasm of the cell, so we're not into the mitochondria yet. We're still out in the cytoplasm. And it's an anaerobic process. No oxygen is needed. There's not a surplus of oxygen in the cytoplasm, and that's okay because it's not needed for glycolysis. 2 molecules of ATP will be produced per every pyruvate molecule. So we have 1 glucose producing 1 pyruvate and 2 ATP. This is the pyruvate molecule that is going to be converted to acetyl-CoA during the transition step before entering the mitochondria so that it can be used in the Krebs cycle. Again, that's happening right here. Pyruvate becomes acetyl-CoA. When we get into the Krebs cycle, we are harvesting energy from that acetyl-CoA that used to be pyruvate, and this is taking place in the inner membrane of the mitochondria. It is an aerobic process. Aerobic, meaning oxygen is needed. There are 2 molecules of ATP produced and several NADH and FADH2, which are going to be needed to make the majority of the ATP in the next step, which is in the electron transport chain. So we've gone over glycolysis and the Krebs cycle process. Here we are producing a byproduct of CO2, the 2 molecules of ATP, and the FADH2 and the NADH that are going to be funneled to the electron transport chain. These are extremely useful because they are working as electron acceptors. In the process of cellular respiration, the glucose is entirely consumed. The energy from its chemical bonds has been transformed into 4 ATP molecules, 10 ADH electron carriers, and 2 FADH electron carriers. So this is before we get to the electron transport chain. When we've done glycolysis and the Krebs cycle, we have these products. These electron carriers are going straight to the electron transport chain to make the majority of our ATP. So here we are talking about the electron transport chain, and you can see we are transferring electrons with redox reactions to provide ATP. This is taking place in the mitochondrial matrix as well, more specifically the inner mitochondrial membrane. It is also aerobic, meaning oxygen is needed for this process to take place. And like we said, the majority of our ATP is coming from this process. We are getting 34 molecules of ATP produced by this. We are receiving our NADH and our FADH2 from the Krebs cycle, and 10 molecules of ATP are going to be made for every NADH molecule, and 2 molecules of ATP are going to be made for every FADH2 molecule. You won't need to know those specific numbers of how many molecules of ATP per NADH and per FADH2, but it is useful when you're trying to remember, okay, what is needed in the electron transport chain? We have to have NADH, we have to have FADH2. That's the main point here. So glycolysis is a sequence of reactions that form a biochemical pathway. You remember we talked about biochemical pathways, and in 10 enzyme catalyzed reactions, the 6-carbon sugar glucose is broken into 2 3-carbon pyruvate molecules. The breaking of the bonds is what's yielding energy that is going to be used to phosphorylate ADP to ATP. And this is that substrate level phosphorylation. In addition, our electrons and hydrogens are donated to NAD+ to form NADH, so it can be an electron carrier. When oxygen is available, the cell harvests pyruvate's energy in 2 steps. It oxidizes pyruvate to form acetyl-CoA, and then oxidizes acetyl-CoA in the Krebs cycle. So here's kind of a zoomed out version. I like this because you can see here's the outer membrane of the cell. This is our phospholipid bilayer protecting the cell from everything outside it. Here is our cytoplasm where glycolysis is taking place. We have 1 glucose molecule creating 2 pyruvate and 2 ATP. As we enter the mitochondria, that pyruvate is transformed into acetyl-CoA. so that it can go into the Krebs cycle. The Krebs cycle will produce our NADH and FADH2, as well as the byproduct of CO2, and then our electron carriers, NADH and FADH2, can enter the electron transport chain and really get to work making the majority of our ATP. When pyruvate is oxidized, a single of its 3 carbons is cleaved off by the enzyme pyruvate dehydrogenase. So here's that cleaving that's happening. The enzyme is coming in and providing an opportunity for this to break off from the pyruvate molecule. The carbon will leave as part of the CO2 molecule, which is a byproduct. And in addition, a hydrogen and electron are removed from pyruvate and donated to NAD+ to form NADH. So here you can see NAD+ received an electron and became NADH. The remaining 2-carbon fragment of pyruvate is then joined to a cofactor called coenzyme A. So here they join together, coenzyme A, which is also seen here. And our final compound is acetyl-CoA. Once we have acetyl-CoA, we have what we need for the Krebs cycle to function. The fate of acetyl-CoA depends on the availability of ATP in the cell. So once pyruvate is turned into acetyl-CoA, if there's plenty of ATP in the cell and we don't really need energy, then we are going to store it so that we can use it later. So if we have plenty of ATP, acetyl-CoA is going to be diverted outside of the mitochondria to the fat synthesis process to make fat so that we can store that energy for when it is needed. But if there's not very much ATP in the cell, acetyl-CoA enters the Krebs cycle in the inner membrane of the mitochondria to make ATP. So what is the main goal of glycolysis? What happens in the transition step between glycolysis and the Krebs cycle? And what is the main goal of the Krebs cycle? Also, what is the main goal of the electron transport chain? The main goal of glycolysis is to transform that glucose into the usable pyruvate. In our transition step, pyruvate is transformed into acetyl-CoA. And really the main goal of the Krebs cycle is to transform NAD+ to NADH and FADH to FADH2 because we need electron carriers. So once those transformations have taken place, we can go to the electron transport chain and make our heavy amount of ATP, where we have 32 molecules. In chemiosmosis, we talked about how mitochondria use this process to make ATP. As the concentration of protons builds up in the inner membrane space, the protons will re-enter the matrix via ATP synthase channels, and their passage powers the production of ATP from ADP. So remember that chemiosmosis is the process we're doing to make ATP when we're phosphorylating it from ADP. Chemiosmosis is what's happening in the electron transport chain. Sugars are broken down in cellular respiration into ATP, carbon dioxide, and water. What are proteins broken down to? And by what process? What are fats broken down into? And by what process? It's not like sugars are the only thing we're ever breaking down. So when we break down proteins, what do we get? And how are we doing that? When we break down fats, what do we get? And how are we doing that? When we're breaking down proteins, this is deamination. And proteins are broken down into their individual building blocks, amino acids. So all of our proteins are made up of amino acids. When we go through deamination, we are breaking those proteins down to the building blocks that they're made up of, amino acids. What are fats broken down into and by what process? Beta oxidation is how we're breaking our fats down. And we're breaking them down into their building blocks as well, fatty acids. So in chapter 8, we went over mitosis, which was a pretty heavy chapter, but it can be pretty easy once you have gone through it repetitively and you can notice the similarities that are happening throughout the process. So cell division in prokaryotes takes place in 2 stages, which together make up the simple cell cycle. First, you have to copy the DNA, and this process is called replication, and then you have to split the cell in 2 to form daughter cells. This process for prokaryotes is called binary fission. So in our prokaryotic cell production, we have replication, where we're copying our DNA, and this is where you remember we had a point of which was called our origin of replication. This is where we are unzipping our DNA so that a copy can be made, and then we have two copies of that same DNA information in the cell. That is what's happening in replication. We are replicating the DNA. In binary fission, this is the process of creating those 2 daughter cells. So you can see here is where our DNA is being replicated in this step of binary fission. And the goal is 2 clone cells. So this is asexual reproduction. We are not interested in combining new versions to get new genetic information in our offspring. We want clones. This is prokaryotic, asexual cell production. So binary fission copies the DNA, and then this cell will elongate and split, and then we have 2 clone daughter cells that have the same information that the parent cell had. So our prokaryotes have that circular ring of DNA, and the 2 daughter cells are clones. This is important to remember. For asexual reproduction, there's nothing to change the genetic information from what the parent provided. So the daughter cells will always be clones. Eukaryotic cells, much more complex organisms, have much more complex ways of reproducing. And in mitosis, this is our cell division mechanism that occurs in our non-reproductive cells. So these are our somatic cells. And anytime that we need to heal a cut or create new cells in our esophageal lining, or heal something that happened to our eye, we have to make more cells that are the same as the ones that were injured. Also, if we're growing, we need to make more of the same cells in the skin. So we are copying those skin cells. We need to make more of the same cells in our lungs. So we're copying those lung cells. And that's why we want our clone cells, because we don't want to have suddenly an eye cell that is growing on our arm. You could just use that visual to remember. Somatic cells are non-sexual cells. That's why we need mitosis, an asexual form of cellular reproduction, to create clone cells, because we don't want any genetic changes between the skin cells and the new skin cells we have to reproduce. In meiosis, this is where it gets way more interesting, because this cell division is the mechanism to basically further our whole species, and we need genetic variability in order to be successful organisms. So in meiosis, this is cell division that occurs in cells that participate in sexual reproduction, and we call these cells germ cells. The eukaryotic cell cycle of mitosis is divided into distinct phases. Interphase, which is where the cell is spending most of its time. Prophase, metaphase, anaphase, telophase, and cytokinesis is happening at the end of telophase. That is when our cytoplasm is splitting so that we actually have 2 separate cells. In interphase, this is the stage before mitosis. It is comprised of a resting phase, and that's where no cell growth is happening. No division of or replication of DNA is happening. It's just kind of the hangout phase. We call that the GO phase. And then there are the 3 active phases of interphase which follow. The G1 phase is where most of the growth is going to be happening, and most cells spend the majority of their lifespan in this phase. The S phase is where our DNA is being replicated in preparation for cell division. So S phase, you can think we have to make new DNA because our cells are preparing to divide. I once saw a visual that was like, for the S phase, it was an S that then was flipped in a mirror image so that there was another S next to it. And that was just kind of a helpful visual that helped me remember that DNA replication is happening in this phase, S phase of interphase. At the end of interphase, we have our G2 phase, and this is where we're continuing to prepare for cell division. This is where we're replicating our mitochondria, and we're starting to make those microtubules that will make up our spindle fibers that we need during mitosis. So once we're in mitosis, please forgive the formatting problems. I had to use a different computer so I could use this recording software, and it seems to have slightly rearranged things, but let's move past that. Mitosis, we have PMAT. Remember that's what we are using to remember the order of these steps. So PMAT, prophase, metaphase, anaphase, and telophase. In prophase, we are preparing. So our chromosomes are condensing, that nuclear envelope is breaking down, and our spindle fibers are forming from centrioles in animal cells. In metaphase, that's where we want to think middle, where all of our chromosomes have got to line up in the middle. That middle that we're talking about is the equatorial plane. They also sometimes call it a metaphase plate. And our spindle fibers are binding to the centromeres. So that process where the spindle fibers made of microtubules are reaching out to bind to the chromosomes, that's happening in metaphase as well. In anaphase, we want to think away. Our centromeres divide and our spindle fibers begin to break down. So the centromeres holding our chromosomes together are dividing so that the separate chromatids can go to opposite sides of the cell. The spindle fibers are breaking down, and that's actually starting at the opposite ends of the cell. That's where the breakdown of those spindle fibers is happening. And that motion of those breaking down from the end of the cell is what actually pulls the chromatids towards the opposite poles. In telophase, you could think, tell everybody we are about to split, because chromosomes are uncoiling, a new nuclear envelope is being formed, the nucleolus is reappearing, and our spindle fibers are disappearing. So you can tell that we are really about to cut in half with cytokinesis because our chromosomes have uncoiled, we've got a nuclear envelope again, the nucleolus is back, and we don't have spindle fibers because they've already completed their job and disappeared. So we can enter cytokinesis, where the cytoplasm will divide and we will have two successful daughter cells that are clones. Cytokinesis occurs at the end of mitosis. It is the division of the cytoplasm into roughly equal halves. So it's not perfectly equal, but it is very close. In animals, cytokinesis occurs by actin filaments contracting and pinching the cell in two. And this action is evident as a cleavage furrow that appears between the daughter cells. You can see it here. When you start to see this pinch, that's the cleavage furrow that is indicating, oh, this one cell is about to become two cells. In plants, when you're looking to try and see, are these cells going through cytokinesis, are they about to divide, you can observe the production of this cell plate, which is basically what will be a new cell wall between the two separated cells. But it's not quite yet, so we call it a cell plate. The cell wall grows at right angles to the mitotic spindle and is called the cell plate. So this is in plant cells. Animal cells, we have a cleavage furrow. Plant cells, we have a cell plate. So here's that breakdown. Here's interphase, where our chromosomes are extended and in use during the G1, S, and G2 phases. The GO phase is just the hangout. It's not mentioned here, but that's where nothing's really happening. We're not growing. We're not replicating. The S phase is where you want to remember we are replicating our DNA. G2, we are starting to get those mitochondria and forming microtubules so that they can be used during mitosis. We enter the beginning of mitosis with prophase, where our chromosomes are condensing, the nuclear envelope breaks down, and the spindles are forming. You can see our chromosomes are just kind of spread out wildly still. And they align in the middle on the equatorial plane during metaphase, our middle phase, where our chromosomes want to be in the middle. So the chromosomes are going to line up on that central equatorial plane, also known as the metaphase plate. In our A phase, we are thinking away from the middle. So here's anaphase. The centromeres divide. Here are the centromeres that would have attached this chromosome if they weren't being pulled apart. So our centromeres are dividing and the chromatids are moving towards opposite poles. That's because these spindle fibers that are attaching to them are being broken down from the pole, and it's kind of reeling in the chromatids to separate ends. In telophase, our chromosomes are uncoiling and we have a new nuclear envelope that is forming. So we don't quite have a fully formed nucleus yet, but that nuclear envelope is forming because we are preparing to split. The spindle fibers are disappearing in this part, in telophase. Then we have cytokinesis, where we have successfully replicated our DNA, moved it to opposite sides of the cell so that we're getting the equal information in each cell that we need. And the cytoplasm is split roughly in half. So our cells that have 2 of each type of chromosome, meaning the chromosomes from mom and from dad, are called diploid cells. And the chromosome number is going to vary among organisms. Most eukaryotes have between 10 and 50 chromosomes in their somatic cells. Remember, somatic cells means non-sex cells, so just all the other cells. Humans have 46 chromosomes and are diploid somatic cells. Homologs, or also known as homologous chromosomes, they contain information about the same traits, but the information may vary. So I mentioned how these could both, they're homologs because they both have the same expression for like hair color, eye color, weight, height, but they could be coding for different things. So it could be, even though they are both talking about the same trait of eye color and eye color here, this could be coding for blue and this could be coding for green. This could be coding for a different kind of hair structure than this, but they, like, one is blonde and curly, one is brown and straight, but they are the same type of gene, just coding for different information. That's what makes them homologous chromosomes. So prior to cell division, each of the homologous chromosomes is going to have to replicate and form 2 identical copies. Those identical copies are called sister chromatids. So once we have 2 copies of this and they are bound by a centromere, that is sister chromatids. They are identical. Those sister chromatids are joined together at the centromere, and humans have 23 pairs of homologous chromosomes, which means that they have 46 chromosomes in each cell. Chromatin are the strands of DNA. So here's our chromatin, this long rope of DNA that is being eventually coiled into our nice, neat X-shaped chromosome. That is chromatin, which is making up our chromosome shape. The chromatin is made up of our DNA strands, and the reason they can be packaged in this nice, neat formation where everything fits together and doesn't get tangled and become a big mess is because of our histone proteins. Our histone proteins have the right affinity to bind to DNA, to bind to our nucleic acids, and bundle up in this nice, neat formation where we have histones that are bundling together into a complex called a nucleosome. So our histone proteins have positive charges that counteract the negative charges associated with the phosphate groups of our DNA, and that's what makes that binding take place. Our DNA is coiling around a core of 8 histone proteins to form a complex called a nucleosome. So imagine that this is 3D and we just can't see the ones on the other side. Here are 4 histones bound together. You imagine whenever it's a complete nucleosome, there are 8 of those histone proteins. And they are all wrapping together nicely so that our genetic information doesn't get tangled and become inaccessible. Also, there are over 50 different proteins that serve as growth factors, and these are the proteins that trigger the cell cycle. So our platelet-derived growth factor, which is often shortened and called PDGF, it is present in blood, and it is what triggers our neighboring cells to divide to heal a wound. So when we get a cut, PDFG is the protein that shows up and says, hey, we need some more cells over here. The cell proliferation signaling pathway binds a growth factor with a specified cell surface receptor. So that's how the signal is received. We have a growth factor that is landing on a cell surface receptor, and that is what triggers an intracellular signaling pathway, and then activates the proteins which are going to initiate cell division. Here's that growth factor landing, and that is like a flip of a switch. Oh, the growth factor's here. It is time for us to enter this stage where we are going to replicate our DNA and replicate our cells. Also, cancer is a growth disorder of cells. It begins when apparently normal cells are starting to grow uncontrollably. The result is a growing cluster of cells called a tumor. Malignant tumors are invasive, and cells from malignant tumors can metastasize, leaving and spreading to different areas of the body to form new tumors. This process is called metastasis. So when we're talking about metastasis, we're talking about how there were cancer cells just growing in one part, you can imagine, in the knuckle of your finger. But when metastasis happens, there's been such a replication of cancer cells to the point where there are so many that now they have entered the bloodstream and they are able to travel to a different part of your body and replicate. So metastasis is when our cancer cells are spreading from where they started to new areas of the body to form new tumors. Here you can see our normal cells, and these are the tumor cells that just keep dividing even though they shouldn't. They should not keep growing and they should not keep replicating, but they are, and that is what is forming our tumors. Cell division is regulated by proteins called growth factors, and cancer is caused by damage to genes that encode those growth factors. So you can imagine if the genetic instructions gets damaged, and it's the genetic instruction for growth factors, which are in charge of telling us when to make new cells, you can see how that correlation makes sense. The mutation causes damage to the genes, and this mutation could come from chemicals, viruses, or environmental exposure to things like UV rays. So our gene mutations for cancer are typically coming from chemicals, viruses, or environmental exposure to things like UV rays from the sun. Once that chemical or virus or UV ray has damaged the DNA, often that can damage the instructions for our growth factors. So now we are not getting the right signals of when we need new cells and when we need to stop making new cells. There are typically 2 general classes of growth factor genes that are usually involved in cancer. There's our proto-oncogenes, and these genes are going to encode proteins that stimulate cell division, so our growth factor proteins. And when mutated, these genes become oncogenes and can cause cells to divide excessively. So we're just not getting the right signal. There's no turnoff to that growth factor. They just kept showing up and we just kept making more cells even though we shouldn't have. There's also tumor suppressor genes, and these genes typically turn off cell division in healthy cells when they should not divide anymore. But if these tumor suppressor genes get mutated, then they can also allow uncontrolled cell division. So cancer results from damaged genes failing to control cell division. In about 50% of cancers, the p53 gene malfunctions because of chemical or radiation damage. So the protein the gene encodes does not prevent tumor growth anymore. Another common cause of cancer is a mutated MDM2 gene, which can become overactive, and its protein product suppresses p53. So here we have p53, which has been damaged and can no longer do its job and no longer stops the cell division when it should. And in this one, our p53 gene is fine, but our MDM2 gene has been mutated, that so much to the point that now it's getting in the way of p53. Almost imagine the MDM2 gene sitting on top of p53 and saying, I won't let you do your job. In both cases, the p53 protein wants to stop cell growth that is leading to tumors, but it can't because of the mutations to its genetic information. That's mitosis. Now let's go over meiosis. So mitosis is the process of making new somatic cells, and meiosis is the process of making new sex cells. The difference is that mitosis is the process of making new cells that are clones of the parent cell, like when we're making new cells to heal a cut. But meiosis is about making new cells for the purpose of sexual reproduction. And this process introduces new gene arrangements in chromosomes so that the offspring will have genetic variability instead of being clones with identical genetic makeup. So we have interphase, which is where the cell is growing, replicating its DNA, and preparing for cell division. What we just talked about was somatic cells. So we have interphase, which comes before a somatic cell enters mitosis. Interphase is also happening in sex cells, though. If it's a germline cell, which is a sex cell, then the process that follows interphase will be meiosis. This should not be here. Sorry about that. But it is also relevant because mitosis ends up with cloned diploid cells and meiosis ends up with haploid cells that are not clones of the parent cell. So our gametes are reproductive cells. That's just the word for our cells like eggs and sperm, and they are the cells that contain half of the chromosomes found in somatic cells. So they have half the genetic information that the rest of the cells in a body have. The gametes fuse during fertilization to form a new cell called a zygote, which contains 2 complete copies of each chromosome. So fertilization is happening here. Half of our genetic information coming from dad is combining with half of the genetic information that is coming from mom. And at that point, a zygote will form, which just means it's now a diploid cell that has genetic information from both mom and dad. And that fertilization process can also be called syngamy. So here, interphase is going to the process of mitosis, which will produce our cloned diploid cells. The cloned diploid cells have both sets of genetic information that came from both of that organism's parents. Interphase also comes before meiosis in our germline cells to create genetically altered haploid cells. So they will have half of the information. They are haploid cells. They have half of the information of the parent cell. When we're thinking no new genetic changes have been made, but new cells have, that's asexual reproduction. Because a, when you put it in front of a word, means without. So without sexual reproduction. Meiosis is a form of sexual reproduction because we have new genetic combinations in our daughter cells. They are not clones. Some organisms reproduce by mitotic division and do not involve gametes at all. That is asexual reproduction. The example that we have here is binary fission, which is happening in prokaryotes. You remember these are clones, so that is asexual reproduction. But there are also organisms that are able to reproduce both using sexual and asexual reproduction. Strawberry plants are able to reproduce sexually by combining new genetic information and going through the process of fertilization with their blooming flowers, but also they're able to send out runners mitotically, which means through mitosis, by creating clone cells and then having clone daughter plants as well. So this has clone genetic information to the original parent plant. But whatever new plant comes after this flower has bloomed, that could possibly have a slight difference because of that gene rearrangement that's happening in meiosis. So our germline cells are what make the gametes. In animal cells, the cells that will eventually undergo meiosis are saved early on for the purpose of reproduction. And these cells are in the ovaries and testes in humans, and they are referred to as germline cells. These cells are diploid, like our somatic cells, and that means that they have the genetic information from both parents. Only the germline cells are going to undergo meiosis to produce our haploid gametes. So if you're trying to think of germ cells and what they mean, you could try to remember that the term germ cells is actually just coming from the Latin word germen, which means like seed or sprout. I put this picture here so you could think like, the seed or sprout of a family tree. Are germ cells. They are the beginning of the genetic variation that happens through our reproduction, our sexual reproduction. So we have germline cells, which are diploid, and those are hanging out in the ovaries and the testes, waiting to make our gametes, which are haploid and needed for sexual reproduction. So in interphase, that's where our DNA is replicated, and that is happening before meiosis begins, just like it happened before mitosis began. So we have a germline diploid cell that has information from both of our parents. And then once interphase is complete, where we have already replicated our DNA, replicated our mitochondria, started to form our microtubule spindle fibers, then we can enter meiosis 1. And that the main goal of meiosis 1 is to separate the homologous chromosomes. So because we have to take this diploid cell, 2N, and make a haploid cell, 1N, we have to pull the chromosomes to opposite sides of the cell so that we can enter meiosis 2, where we will then actually separate the chromatids from each other. So this picture looks a lot like mitosis because we just have one line of chromosomes in the middle and they will be pulled apart at the centromere to opposite ends. That's not happening the same way in meiosis 1 because we have to end up with half the genetic information. So that's why we are pulling one whole chromosome comprised of 2 chromatids to this side and 1 to this side, so that we can end up with 2 haploid cells. But 2 haploid cells is not the goal of meiosis. 4 haploid cells is the goal of meiosis. So here we get a lot of our genetic variability, and then we need to take that genetic variability and split it again so that we have, that's not really the best way to say it. Here is where our genetic variability is coming into place because we are separating homologous chromosomes, and that's where our crossing over happens. And we have independent assortment that is bringing in all of our genetic variability in meiosis 1. But we don't want to end up with just 2 cells. We want to end up with 4 daughter cells. So now we need copies, and the copies are made in meiosis 2. They're still not going to be clones though, because you can see different chromosomes got pulled to different sides. So then when these split and go to different sides, it's not like these are all going to end up, it's not like two of them will be the same as each other. All four of these individual gametes will have separate genetic information because of that chromosome division. And because of the different crossing over that happened as well. Whoa. This slide should not be here, so we're going to just scroll on by. Meiosis 1 is divided into 4 stages. We've got prophase 1, which again, we are prepping. We are preparing for mitosis. This is the beginning. Our nuclear envelope is breaking down. Our homologs are pairing up and exchanging segments of chromosomes through crossing over. So that's where a lot of our genetic variability comes from, in prophase 1. That's where crossing over occurs. You remember when the chromosomes overlapped and exchanged little pieces of themselves so that they could actually be more diverse? That's what's happening in prophase 1. In metaphase 1, again, we're going to the middle, but instead of it just being a line of single chromosomes, it's actually a line of double chromosomes up and down the equatorial plane of the cell. The random orientation of the chromosomes, as some of them end up facing the north pole of the cell and some of them end up facing the south pole of the cell, is also going to provide more different combinations of parental chromosomes. So that is what's called independent assortment. And that's just the variability that comes from the different orientations of which way those chromosomes are facing before anaphase when they're pulled away. So anaphase, we have away, where we are going away from the middle. And anaphase 1 means that we're pulling a whole chromosome made of 2 chromatids to each side of the cell. So this whole chromosome with 2 chromatids is being pulled to one side. This whole chromosome with 2 chromatids is being pulled to one side. And our homologous chromosomes are being pulled all the way to the opposite ends. So telophase 1, we are telling everybody that we're about to split. This cell is almost done. Our DNA is being replicated before meiosis begins, during interphase. So just like interphase came before mitosis, interphase comes before meiosis. This is where the DNA replication is happening, in interphase. So here's our diploid germline cell, whose goal is to produce 4 haploid cells called gametes. In meiosis 1, the main goal is to separate our homologous chromosomes. So pull these chromosomes to opposite sides of the cell. We're not pulling chromatids to opposite sides of the cell. We're pulling chromosomes with 2 chromatids to opposite sides of the cell in meiosis 1. You could think meiosis 1, you're taking one whole chromosome to an opposite side of the cell. Meiosis 2, the main goal is to separate the replicate sister chromatids. So these are replicate chromatids of these chromosomes, and they are being pulled, each chromatid is now being pulled to opposite sides of the cell. When meiosis is complete, the result is that one diploid cell has become 4 haploid cells. Has produced 4 haploid cells is probably a better way to say that. So again, think about interphase. The steps remain the same if it's coming before meiosis. We have the GO phase where no growth is happening, the G1 phase where the primary growth phase of the cell, this is the primary growth phase of the cell following division. Most cells spend most of their lifetime in the G1 phase. The S phase is when your DNA is replicating because you're preparing for cell division. So whenever a germline cell needs to start making gametes, that's when the S phase of interphase happens, because DNA replication will begin to occur to prepare for that cell division. In the G2 phase of interphase, there's more preparation before meiosis, which includes replication of mitochondria and synthesis of microtubules. So then it's time to enter meiosis. And meiosis 1, which is the first half of meiosis, It is divided into 4 stages, conveniently with the same name as the steps that we had in mitosis. So we've got our PMAT, where prophase 1 is where we're prepping. Metaphase 1 is where our homologous chromosomes are coming to the middle. Anaphase 1, those chromosomes are being pulled to opposite sides of the cell, opposite poles of the cell. And telophase 1, or you could think, tell everyone we're about to split, because the chromosomes are gathering at each of the 2 poles before cytokinesis. All of these steps are called prophase 1, metaphase 1, anaphase 1, and telophase 1 because there's a whole second half of meiosis called meiosis 2, and those steps will be called prophase 2, metaphase 2, anaphase 2, and telophase 2. So here's our diploid germline cell. The chromosome duplication takes place, an interphase. Then meiosis 1 provides a lot of genetic variability. We have crossing over with our chromosomes that provides genetic variability, as well as independent assortment, which we should talk a little bit more about, I realize. Let me go back to that last slide. So in prophase 1, our nuclear envelope is breaking down. The homologs are pairing up, and they're exchanging segments of their chromosomes via crossing over. So you remember, this is when the chromosomes kind of overlapped each other and traded segments so that each chromosome could end up more diverse than they were before the crossing over. After that, when we're in metaphase, our pairs of homologs are aligning in the middle of the cell on the equatorial plane, and we have pairs, so it's 2 chromosomes, 2 chromosomes, 2 chromosomes down in a line. And actually the random orientation of those chromosomes, as some of them end up facing the north pole of the cell and some of them end up facing the south pole of the cell, is going to result in gametes with more different combinations than the parent chromosomes. So that process of providing genetic variability is called independent assortment. When you think independent assortment, think it is in metaphase, and it's just about the orientation that the chromosomes land in when they get to that middle of the cell. In anaphase 1, we are thinking away. We are going away from the middle. We are pulling these chromosomes away to the opposite poles. In telophase 1, we're thinking, tell everyone we're about to split, because the chromosomes are all gathering at the opposite poles of the cell before cytokinesis happens. After telophase 1, cytokinesis happens, and now we have 2 daughter cells. But the goal is 4 daughter cells. Meiosis 2, so now that the genetic variability is taken care of by crossing over and independent assortment that happened in meiosis 1, meiosis 2 just needs to make 4 cells from the 2 cells that resulted from meiosis 1. So we enter meiosis 2. It is also divided into 4 stages: prophase 2, metaphase 2, anaphase 2, and telophase 2, which should be becoming pretty familiar at this point. In prophase 2, we are prepping again. So our new spindle fibers are forming to attach to the chromosome clusters at the opposite ends of the cell. In metaphase 2, we are going to the middle again, except this time, instead of a line of 2 chromosome pairs, it is just individual chromosomes aligning in the middle. That middle is still called the equatorial plane. And in metaphase 2, the spindle fibers are going to bind to both sides of the centromere, because we need to split that centromere in half to end up with individual chromatids on either side of the cell. Anaphase 2, we're thinking away. Our sister chromatids are pulled away to the opposite poles. You can see here, this chromatid will be pulled over here. This chromatid will be pulled over here. And in telophase 2, tell everyone we're about to split. The nuclear envelope is reformed around each of the 4 sets of daughter chromosomes. So now we've got a nuclear envelope back. The copies of genetic information are protected inside that nucleus, and cytokinesis can take place for a second time. So here's our second cytokinesis, where these cells have got all the chromosomes to opposite poles, and that cleavage furrow or cell plate has formed, and now we have 4 haploid gametes. So that crossing over in prophase, here's a picture to show it happening. It's here we have adjacent homologs, which are non-sister chromatids. So these are not identical. They cross over and they trade so that they can each have more genetic variability. And this says, think recombinant chromosomes, like recombined chromosomes. So what is produced by crossing over in prophase 1? Recombinant chromosomes, which is chromosomes that have been recombined with a new genetic information combination. So how did we generate the new genetic combinations during meiosis? There were basically 3 things that resulted in all of this genetic variability in the offspring cells, then the daughter cells. First was crossing over during prophase 1, which we just talked about. Second was that independent assortment of just the random orientation of the chromosomes as they align in the middle of the cell during metaphase 1. And also just the randomness of whenever fertilization happens, because which sperm reaches which egg is going to vary depending on when the fertilization actually takes place. So that also provides even more variability. Okay, and alleles and natural selection. Also, I just want you to remember that an allele is one of 2 or more alternative forms of a gene that can arise by mutation and are found at the same place on a chromosome. When changes in the environment occur such that a certain allele provides a benefit to the individuals that carry it, the natural selection is going to act to favor that allele, so that it can get passed on to future generations. So think about that. If you have a gene that has alternate forms of it, so a color gene that could either code for lighter expression or darker expression on a moth, whichever one is going to provide a benefit to those individuals, that's what's going to be favored. So when we're favoring that allele, that means it is preferred to be passed down to daughter cells and eventually to future generations of that species. The example that we talked about were the peppered moths that were affected during the industrial boom, when all of the trees became covered in soot, and suddenly that gene expression for a darker pigmentation became favored because that would let the moths survive better in the new conditions of the soot-created world that came from all of the industry and all of the production that was happening from humans. So heredity is just the sum of all biological processes by which particular characteristics are transmitted from parents to their offspring. So basically, heredity is just talking about how did we get what genes from each of our parents? What did we inherit from the people that created us? And a trait is an expression of that characteristic. So a trait could be something like hair color, height, eye color, the expression of what you've inherited genetically. We talked about Gregor Mendel. He is known as the father of genetics due to his success in the study of heredity using experiments with different varieties of pea plants. You remember he was the monk in the 1800s who did numerous pea plant experiments. In those experiments, Mendel self-fertilized different varieties of pea plants to create true breeding parental generations for his experiments. And that true breeding just means that he was creating lines so that if they were bred with each other, all of their offspring would have the same phenotype as the parents. So basically, he noticed that there are pea plants in the garden, they often are purple. Sometimes they randomly come up white. And then he took some of the purple flowers and some of the white flowers, and he kept self-fertilizing them, so no new genetic information was introduced. He kept self-fertilizing them until, no matter what, the purple flower plant always produced purple flowers, and the white flower plant always produced white flowers. And that was what was called the true breeding generation. So he named that true breeding generation the parent generation. So once he had done that enough to where he was like, okay, these are always coming out purple now, that's whenever he said, I've got a true breeding parent generation of purple or white. The offspring from a cross of true breeding parental genotypes is known as the F1 generation. So whenever you have, you're mixing these two true breeding varieties, and you end up with a new generation. That's called the F1 generation. You remember that F just stood for filial, which just basically means offspring. So the F1 generation is then used and self-fertilized so that the offspring from two heterozygous members of the F1 generation are known as the F2 generation. So we have, two F1 plants. They're in the same generation, but two separate F1 plants are bred together, and that creates the F2 generation. Mendel proposed a simple set of 5 hypotheses to explain his results. In hypothesis 1, he said that parents do not transmit traits directly to their offspring. Parents transmit information about the trait in the form of what Mendel called factors. So he's saying daughter plants are not just inheriting the color purple from their parents. They're inheriting the information that codes for the color purple, which he called factors and which we now call genes. Hypothesis 2 was that each parent contains 2 copies of the factor governing each trait, and the 2 copies of the factor may or may not be the same. So he said that homozygous individuals are individual chromosomes that have the same copies of a gene. So here's a homozygous chromosome. These are coding for the same thing, and that is what makes them a homozygous. As opposed to heterozygous chromosomes, where these are about the same trait, but they're coding for different things. In hypothesis 3, as a result of this hypothesis, alleles were discovered and named about 70 years later. Alleles are one of 2 or more alternative forms of a gene that can arise by mutation and are found at the same place on a chromosome. So they're coding for the same thing, but they could be coding for, they're coding for the same thing like eye color or plant color, but that information could differ. That's what an allele is. The appearance is determined by the alleles a plant receives from its parents, and the alleles a plant receives from its parents is the plant's genotype. So whatever alleles you receive from the plant parent, that's the genotype. But the expression of those alleles, however they show up in a way that you can observe, is the phenotype. So our genotype is just the genetic makeup, the genes that we got from our parents. And it's what determines the phenotype, because the phenotype is just the physical expression of those genes. So genotype says, what genes did you get from your parent? And phenotype says, how are those genes expressed? You can see that our homozygous plants that have a purple color could either be heterozygous, which means they had different copies of those genes, or homozygous, where they had the same copies of those genes. Also, you can see the phenotypic expression of this plant. It is a homozygous plant. It has 2 copies of the same gene. And the expression, the physical expression or phenotype, is the white color of the flower. In hypothesis 4, Mendel said the 2 alleles that an individual possesses do not affect each other. So they're separate. Their coding and expression is separate from each other. They don't affect each other. That was his 4th hypothesis. His 5th hypothesis was that the presence of an allele does not ensure that a trait will be expressed in the individual that carries it, which we see all the time in the heterozygotes, because often only the dominant allele of those 2 mismatched alleles is what is expressed. So our dominant traits are capitalized, while a lowercase letter is reserved for the recessive trait. So on those heterozygous alleles, you saw that some of them had a capital P and a lowercase p. The dominant allele is the one that is more likely to be expressed. So even though they're both there for a heterozygous individual, only the dominant allele is being expressed. When 2 heterozygous pea plants are crossed, the genotypic ratio of the offspring will be 1 to 2 to 1. You remember in class we talked about how at first he thought it was a 3 to 1 ratio, because in the F2 generation he often saw there are 3 purple flowers for every 1 white flower. But as he studied this further and he got more and more data and did these experiments over and over again, he came to realize that it's a 1 to 2 to 1 ratio that kind of looks like a 3 to 1 ratio. But that's because we have our heterozygous offspring here. They have genes for both the purple and the white expression. But because the purple expression is dominant, more often than not, we're ending up with a 3 to 1 ratio. But it's not that simple, because what we really have is the possibility for purple in 2 of our offspring. And then 1 of those 4 will always be purple because it is homozygous dominant. It has both of the dominant genes that match each other, that both code for purple. Then 2 of the 4 we have that could possibly be purple, but are most likely going to be purple, but could also express that recessive gene and end up being white. And then 1/4 that will always come out white because they got the 2 recessive white genes that are expressed. There's no purple gene present to code for that purple color. And the results from a cross between a true breeding white flowered plant and a true breeding purple flowered plant can be visualized with a Punnett square. So this is what he came up with so that he could try and get a better understanding of what was happening with these generations. A Punnett square lists the possible gametes from one individual on one side of the square. So here's one of the parent flowers, and lists the other parent in a procreation on the top side. And this is how you're able to see that all of the possible gametes that could result in the offspring. The genotypes of potential offspring are represented within the square. So here you can see if we cross our F1 heterozygous flower with another F1 heterozygous flower, then here's our 1 to 2 to 1 ratio. This 1/4 will end up dominantly, it's a homozygous dominant genotype, so it will express purple no matter what. there's no lowercase recessive white gene here. So it will, 1/4 will definitely be purple. 1/4 will definitely be white because there's no dominant gene present. There's only the two homozygous recessive genes. And then in 2 of the 4 offspring in the ratio, these could end up being either purple or white, but more often than not, they will be purple because they have a dominant gene there that is coding for purple. So that's what makes our it's our 1 to 2 to 1 ratio. And here you can see that with the visuals. So this is our parent generation true breeding for white, our parent generation true breeding for purple. Whenever we cross those together, all of the individuals in the next generation are going to have the same genotype because they are mixing one dominant purple allele with one recessive white allele, and they all have the same genetic coding. But once we cross 2 of those heterozygous species from the F1 generation to make the F2 generation, that's where we get our 1 to 2 to 1 ratio for the offspring of and the phenotypic expression that we see there. So in Mendel's hypotheses, they were so neatly predicting the results of his crosses that they've actually, some of them have come to be known as Mendel's laws. And Mendel's first law is the law of segregation, where he said the 2 alleles of a trait are separate from each other during the formation of, are separating from each other during the formation of gametes. So that half of the gametes will carry one copy and half will carry the other copy. So he's saying these 2 alleles that code for the same thing are being split up during the formation of the gametes. And in the second law, Mendel's second law, his law of independent assortment, he said genes located on different chromosomes are inherited. Oh, sorry about that. Genes located on different chromosomes are inherited independently of one another. So this law states that whatever genes are located on different chromosomes, they're going to be inherited independently of the other genes. They're not thinking about, well, what gene did you get? Do you really need me? No, they're just separate from each other in their inheritance. Also, when multiple genes are acting together to influence a phenotypic expression, that is a polygenic expression. So examples include height, eye color, and weight. These are all the same phenotypic expression, but multiple different genes are affecting them. And that is called polygenic expression because you're using more than one gene to affect a phenotype. On the other side, when you have one gene that has multiple different effects on a phenotype, that is called pleiotropic. So if you have one gene that is affecting multiple different traits, that is a pleiotropic gene. And the example we talked about in class was the CF gene, which produces faulty chloride transport channels and causes mucus to build up in the liver, pancreas, and lungs. And this is just one single gene, but it has all of these negative effects. It causes blocked fat digestion, resulting in vitamin deficiency. It causes failure of cilia to flush mucus from the lungs, causing respiratory infections, and also causes salty sweat. So just one gene causing all these different things, that is a pleiotropic gene. In chapter 10, they also discussed Thomas Hunt Morgan's work with fruit flies. And in 1910, Morgan discovered a mutant male fruit fly who had white eyes instead of the typical red eyes. He was trying to figure out whether this trait would be inherited by the Mendelian pattern that Gregor Mendel proposed. He crossed a mutant male with a normal female. So the normal female has red eyes. And as predicted, eye color segregated, and all the F1 individuals had red eyes. But in the F2 generation, the white eyes only showed up in males. So Morgan determined that sex was the key to explaining the results of his cross. In fruit flies, sex is determined by the number of copies of a particular chromosome, the X chromosome, that an individual possesses. A fly with 2 chromosomes is female, and in male flies, there's only one X chromosome, and that is paired with a Y chromosome. So Morgan reasoned that the white-eyed trait had to reside only on the X chromosome. And that meant and that led to the revelation that a trait could be sex-linked. It could only show up on the genes that determine sex.